All right, everybody, how we doing? Welcome back. Thanks for tuning in. Today we're going over our third lecture over cells and organelles. So again, we'll be moving through this a little bit quickly. You've seen a lot of this in previous biology classes. If you haven't, or you had a hard time with this information, I highly encourage that you go to SI with your SI leaders, talk to your TAs that we have in class, come into my office hours. We can do a private one through Zoom. We are going to do weekly group ones through Collaborate Ultra. Get this basic information down because really it is foundational for how the rest of the semester works. Understanding what these structures are doing is going to be really important for getting a bigger understanding of the picture. Remember, last lecture, we talked about having multiple perspectives, and so if you're having trouble with this foundational stuff, it's going to make it more difficult to understand how tissues function and these organs function. All right, so to talk about our cell structure here, we're going to basically look at our sales management roles, our training programs, our performance. This is the wrong PowerPoint. Okay, here we go. We're going to talk about cell structure. All right, now we want to look at these organs and their organelles that exist within it. So pause the video here and try to list out as many cellular components that you can remember from previous biology classes, and try to also list out their associated functions. All right. So what are we going to be learning about in today's lecture? So we are going to look at the plasma membrane in good detail. We really need to understand how the plasma membrane works because this is going to be foundational for how neurons transmit their signals. We're going to learn about proteins that are integrated into the plasma so that we can understand how ions move through those proteins. Okay? What is a selectively permeable membrane? What are microvilli and cilia? What do they do? And then we're going to quickly go through the cell structures and the organelles and then go over the functions of those organelles. All right, so if you remember this image from last lecture, our hierarchy of structures in our bodies. And so we're going to be focusing on the organelle and cellular level here. Okay, so cells, these are the functional units of our bodies, and they are enclosed by membranes. There's over 200 different types within our bodies. We're going to be learning a lot about many of these. Hey, Quest, didn't want to leave you off there. All right, so some cell shapes that you should be familiar with. Let me pull up my laser pointer here. And you will be learning about squamous cells. So this means basically flat and squished. We have cuboidal cells that look like cubes. Columnar cells, which are column shaped. And then we're going to also talk a little bit about discoid, fusiform with our smooth muscle. And these three are going to be our main three types that we'll be talking about. But we'll also see a little bit of this and this as well. So I would say if you were going to memorize this slide and memorize these terms, these three you have to know, and these two would be really good to know. All right, so how did you do on your list of structures? Did you name all of these structures that exist within a cell? Can you remember all of their functions and what they do and how they interact? Do you remember the model that you made probably in third grade with like gumdrops and candy and spaghetti? Did you have to do that? So anyways, if not, again, I recommend utilizing the resources that are available to you. SI, office hours, TAs, we are here to help you learn. So we can break a cell down into three main components. First, our plasma membrane. We're going to spend a lot of time learning about that. Then we can look at the cytoplasm and all the organelles that exist within there. And then finally, our nucleus. So if we start with our plasma membrane, what does it do? Well, first, it provides flexible and structural support to our cells. If you remember the difference between plant and animal cells have cell walls, where animal cells have plasma membranes, and so they're not as strong as a plant cell is. So they also serve as transporters, enzymes, receptors, and identity markers. So how does the plasma membrane do that? Well, it does that with proteins that are embedded within the plasma membrane or within the phospholipid bilayer. And those proteins have special functions, as always, form and function. Okay? So let's look at the plasma membrane and look at that phospholipid bilayer. Now, remember, we have these polar heads that are hydrophilic or water-loving, and then we have these non-polar tails that exist that are hydrophobic or water-fearing. So the inside of our phospholipid bilayer is hydrophobic and the outsides are hydrophilic. If we look a little bit closer at that hydrophilic head, you can see we've got our phosphate and glycerol up here, and our hydrophobic tails are composed of those saturated and unsaturated fatty acids. Now, what's really nice about these phospholipid bilayers is that it's really good at keeping things out or inside of a cell. And this is sort of the basis for how soaps work, right? Is we are destroying or breaking those phospholipid bilayers. And if you've ever poured water and oil together, you kind of can see, if you look really closely here, you can see that the hydrophobic heads move towards the water, and those hydrophilic heads move towards the water because they're water loving, and the hydrophobic tails will move towards the oil, and that's going to surround the oil in these little structures called micelles, and that's going to break up those oils and allow us to remove those oils. Okay, now the plasma membrane, or the theory that describes our plasma membrane, is called the fluid mosaic model. And what that is basically based off of these two statements right here, that the proteins within this membrane are distributed unevenly, okay, like a mosaic, right? So here's a really nice picture that's a mosaic, and things are just kind of put together in there to make this really beautiful picture. But our plasma membrane looks sort of similar. We have all these tiny little proteins all unevenly distributed throughout that cell membrane. But then those proteins are fluid. They can move within the membrane, okay? And so they're kind of scattering around and spreading out. Even the phospholipids will actually migrate and spin and move within this plasma membrane, okay? And so this is where this fluid mosaic model comes from. Now, if we look at those proteins embedded into our plasma membrane, we see that they have functions. One, that they offer support to our membrane. They act as transport molecules across the membrane. So you can see this purple one in the center here has a channel through it where some substance can move through. They can cause chemical reactions with other substances that they interact with. And they have receptors for various molecules or hormones, and those are going to trigger some kind of intracellular process. Okay? And then last, they also act as markers. So they help us, or they help our cells identify themselves as self, or identify to the rest of the body who they are, what they do. Now, if we look at those proteins, we can divide them into two categories. They can be peripheral or integral. Okay, so integral proteins span the entire width of our plasma membrane, while peripheral proteins are partially embedded or on the periphery of that internal or external component of that membrane. All right, now here's an image that you're going to want to get some practice with. If we take a look at these different types of proteins, what you can see is here we have a receptor protein. So that protein is going to bind some kind of chemical receptor or some kind of chemical signal. And then we're going to have some kind of intracellular effects. So these might be hormones. Here to the right, we have maybe an enzymatic protein. So we're going to have some kind of chemical reaction here where we're going to take this chemical and we're going to break it down. Our next one may be what's called a leak ion channel. So we're going to have ions that are leaking through these. They're always open or mostly open, and they allow ions to move in and out, while other channels are gated or require some kind of chemical to bind to it or some kind of electrical disturbance to open it or close it. And then we have cell identity markers. So this kind of looks like a little antenna coming up here. And so this would be a glycoprotein. So we have our protein with a glycogen added to it. And this is going to distinguish itself, right? And so that this is a signal to the immune system, especially, and to the body that, hey, I am supposed to be here, okay? And so please don't destroy me. And then this last one over here on the right would be an adhesion molecule. So you can see another plasma membrane right up here, and there's a protein embedded there, and we see the same type of protein embedded here, and they're holding on to each other, and so that's going to help keep our cells connected to each other. So go ahead and pause the video here and answer these questions on your own. Write down some flashcards, and then we'll move on. Okay, so kind of going off of our cell adhesion molecules, right? We want to talk a little bit about cell junctions. So how are these cells held together? Why do they stay there? Why aren't they just floating around in our bodies? So the first type of junction that we have is called a tight junction. So we can see tight junction up here. It almost looks like some quilting or stitching that has held these plasma membranes together. And what that does is it creates a really, really tight junction so that substances can't pass through in between these cells, right? Because we're trying to create a barrier. We don't want things moving in and out freely between the cells. What we want is, if anything is going to pass through, we want the cells to regulate that. So we want the cells to be in control, okay? So as we'll see, this is a little bit loose in later definitions, but it is for the most part impermeable. Our second type is the gap junction. So if you see down here, we have our gap junctions, and these are communicating junctions. So these are proteins called connexins, and they make a little hole or pathway so that ions can move between cells and communicate electrochemically between cells. And then our third type of cell junction we can see up here are these desmosomes. These are anchoring junctions. You can see that we have fibers coming off of these and kind of integrating into the cytoskeleton of our next cells. So this is going to be really strong in anchoring. We'll talk about some in a little bit about what hemidesmosomes are, and those are going to connect our cells at the basal portion to the basement membrane, and we'll talk about what that is doing later. So surface specialization, if we talk a little bit about some structures that we see on the surface of our cells, the first one is cilia, okay? And so these are hair-like structures that are beating in unison. So up in the top right, you can see the cilia of your trachea. And so as you inhale, there is various particulates in the air that get trapped into mucus, and then the cilia help to beat and move that trapped dust and particulates up away from your lungs back into the back of your throat, where you generally end up swallowing it and digesting whatever gets trapped in there. Below that, you can see that those cilia, this is a cross-section of the cilia, they are composed of these microtubules, so these larger structures that help them move and beat in unison. Okay. Our other structure that we'll see a lot of are microvilli, and these are extensions of the cell membrane. So they come off of the cell membrane and they are there to increase surface area. So when we see microvilli, we should be thinking, oh, microvilli, increasing surface area, therefore if we're increasing surface area, we're probably trying to absorb things into our bodies, okay? So surface area is a good indicator of functionality. And microvilli, then, we tend to see these in our intestines, where we're trying to absorb nutrients into our body. Now, just to kind of show you what that looks like, if we take a little section of our small intestine called the jejunum is highly, highly folded, but then when you open it up, you see that there's these smaller circular folds inside of that tube. And if we zoom in a little bit, we see that on those circular folds are villi, or a villus means finger-like, or finger projection. So we have all these little folds on the circular folds, again increasing surface area. And if we zoom in even more, these villi are lined with epithelial cells, in this case simple columnar epithelial cells. All along this border are these tiny little cells that you see to the left, and each one of those cells has microvilli, all these extra folds. So within our intestine, the amount of surface area is astronomical, so that we absorb as many of those nutrients as we possibly can into our blood system, and then our body regulates what's coming in and what's not. All right, so now let's look a little bit more closely at the cytoplasm and organelles that exist within these cells. Now, first distinction is cytoplasm and cytosol. Okay, if you look here, cytoplasm contains all of the organelles plus the fluid that surrounds them, where cytosol is just a term that's referencing just the fluid and not the organelles. So a small distinction, but an important one as you're reading text. All right, so what organelles do we have? Here is a big list of organelles that you're going to want to know how they work and what they do, because they will come back to help your understanding of the body. Okay, so let's talk a little bit about the cytoskeleton. So this is a lattice of microfilaments and microtubules. So you can see in this image here, we've got actin down here on the left, and so I'll pull up my laser pointer here. So actin, we have intermediate filaments, excuse me, and we have microtubules, and these all have different sizes and different functions. These help play a role in cell movements, and they also help to move organelles around, move molecules within the cell. So here's just another image of what that cytoskeleton looks like, just to kind of give you a little perspective of some three-dimensionality here. You can see that here's that desmosome holding a couple cells together, and you can see we've got intermediate filaments that are coming in to our cytoskeleton and kind of meshing in with our cytoskeleton to help make a really secure connection between those cells. Again, we have the same thing going on down here with our hemidesmosomes and that basement membrane. So let's move on and look at each of these filaments. So the first one is actin. Okay, and we call this a microfilament, right? So these are really small in diameter, 7 nanometers. So a human hair is about 0.05 millimeters or 50,000 nanometers. And these guys are 7 nanometers. So just so you kind of have an idea of what's going on. Now these, think of contractin, actin, okay? They're going to help with movement of structures. So in this amoeba down here, we have this little pseudopod, and so as it's approaching some kind of food, these plasma membranes are going to move out and surround the food and engulf that food. And it uses these pseudopods to do that, and the actin filaments are going to help with that movement. We see those actin filaments inside of microvilli. So as substances are being absorbed into this increased surface area, those actin filaments can kind of milk or kind of contract those microvilli to help move those substances down and into our cell. With intermediate filaments, they have some mechanical support, but they're not involved in movement. So they do just provide more of that structural integrity of the cell. And then last is our microtubules, and these are the thickest. They offer a lot of support. You can see here they kind of act as train tracks in helping guide structures around inside of our cells. They also help with motility. If you remember mitosis and the divisions of the mitotic divisions. So there's those spindles that are going to help pull apart those chromosomes. These are microtubules. Okay, next we have lysosomes. So lysosomes, as you can see here, in a couple of lectures, we're going to talk about endocytosis. So what happens is if that cell is taking in some kind of substance, okay, as we can see here at number 1, so some kind of material is coming into the cell, and as it comes in, we have this invagination of that plasma membrane, which then fully surrounds those substances and it buds off, and now we have this vesicle, and inside of that vesicle is some kind of material. Well, these lysosomes, we see we have a primary lysosome because it hasn't bound to that vesicle yet. That primary lysosome migrates its way to this newly introduced vesicle, and when it finds that vesicle, it then fuses its plasma membrane. When it does that, we call it a secondary lysosome. And its digestive enzymes then are released and combined with this vesicle to break down whatever material is inside of there, and then the cell can use that or destroy it and excrete it. But again, you can think of these lysosomes as little digestive packets. Peroxisomes are like lysosomes, except for the fact that they aren't made by the Golgi apparatus. Peroxisome, so you get peroxide, they make hydrogen peroxide. They help break down hydrogen peroxide into water and oxygen, and they'll break down alcohol or free radicals inside the cell. All right, next, good old mitochondria. Hello 181, welcome back. So mitochondria, this is where we see some oxidative metabolism going on here. This is where we're going to see the formation of a lot of ATP, right? Our energy storehouse of the cell, right? So this is our primary energy source is ATP, and our mitochondria are highly involved with making that ATP. So on that note, a few terms that you will need to know. First, glycolysis. So glycolysis, lysis, right? So glyco comes from glucose, and lysis means splitting. So you can see up here, glycolysis is occurring in the cytosol, okay, outside of those mitochondria. And we're taking glucose, we're breaking it down into pyruvate, and then pyruvate is going to enter into our mitochondria, right? And then run through this whole process in making ATP for us. gluconeogenesis. So gluco is making glucose from new sources. So neo is new and genesis is making. So this is our metabolic pathway that's going to generate glucose from non-carbohydrate sources, so from proteins or lipids. And so that can happen in the mitochondria or the cytosol, and it's basically the opposite of glycolysis. So we're basically putting other compounds together to form glucose. Then we have glycogenesis. Okay, so again, glycogenesis, we are going to form glycogen from glucose, and this is where we're going to store it. So when our blood glucose levels are too high, our pancreatic islets are going to release insulin, and we talked about this with homeostasis, and that insulin is then going to trigger our cells to store glucose as glycogen. And then last, we have glycogenolysis, so glycogen lysis. So basically, we are breaking down glycogen into glucose. So this happens in the liver and muscles, and again is regulated with glucagon. Okay, and so glucagon is released from our pancreatic islets, from our alpha cells, and when our blood sugar is too low, we release that glucagon, and that's going to take that stored glycogen and create glucose for our bodies. So make sure you have a good understanding of these four terms. You'll see this a lot in this class. We'll be talking a little bit about this here, and then you'll also see it again in 202. Moving on to ribosomes. So ribosomes, if you remember, there are two subunits of ribosomal RNA, a large one and a small one, and they can be free floating in the cytoplasm, or they can be bound to the endoplasmic reticulum, which then is considered rough endoplasmic reticulum. The ribosomes are there to read the messenger RNA and put together and synthesize proteins based off of those codes. So you can see here our rough endoplasmic reticulum. We've got lots of ribosomes on the surface of our endoplasmic reticulum. And so basically, as those proteins come off, they enter into our rough endoplasmic reticulum, which then modifies them and packages those proteins, and then they're secreted off of that rough endoplasmic reticulum. If you look here, we can see that ribosome reading that mRNA template, creating a protein. That protein comes in, it's modified, and it's packaged into a transport vesicle. That transport vesicle then is sent over to our Golgi apparatus. The smooth endoplasmic reticulum does not have ribosomes on it, and basically has enzymes for steroid synthesis or inactivation. It's producing lipids, helps store calcium, and we'll see that with our muscles. And it also helps to detoxify or inactivate other compounds. And then we have our Golgi apparatus. So we just talked about that transport vesicle here that's coming from our rough endoplasmic reticulum. It's got a modified protein, and we then bring it to our Golgi apparatus. And in the Golgi apparatus, we see that it's modified a little bit more. So adding and removing sugars, and again packaging it so that now it's ready to be sent out and distributed either to the cell membrane or within the cell or externally into the external environment. And so you can see here that this cell has a little protein that is being made that's going to be embedded into the plasma membrane. So that might be a glycoprotein or something. So we've made it through these learning outcomes, and we're going to continue on our journey here. And now what we want to do is talk a little bit about our nucleus. Okay, so what's the structure and function of the nucleus? What are the general organization of genetic material? And what's the central dogma or the central message of molecular biology, right? So if we look at our nucleus, we see that it is surrounded with this nuclear envelope here. We have nuclear pores that are going to allow messages out and information in. And we can look at different cells in our body. So if we look at this skeletal muscle fiber, what you'll notice is that these cells actually are multinucleated. And so in some areas of our body, we need lots of messages, right? We need to make a lot of proteins. So we need a lot of copies of mRNA coming out of that nucleus and going to our ribosomes so that we can have a higher production of proteins. On the other side of the spectrum, though, we have cells like our red blood cell, which actually lose their nucleus because their main function is transporting oxygen. And so they are full of hemoglobin so that they can bind more oxygen, or as much oxygen as possible. So having extra organelles and nuclei is not necessary. They don't need it. And these cells are going to live about 120 days. All they are going to function for transportation, and our bodies are just constantly making new cells every second. So if we look a little bit closer, a couple of things to point out. We've got our nuclear envelope that's surrounding that nucleus. Within that is our nuclear pores, so hormones are going to come in, mRNA molecules are going to move out. And then we usually, on a slide, can see that nucleolus, if you remember that from 181 or previous bio classes. And that's where this dark area that has genes for the ribosomal RNA in it. Inside that nucleus, we can look at DNA and chromatin. Now, if you remember last lecture, I showed you a question where I was asking you about nucleosomes or chromatosomes, right? So this is where this image comes from. So do you need to memorize everything on this slide? No, but what should you know? Well, hopefully at this point you know that DNA is a double helix. That's important. You should know the base pairing. ATGC, that's really important. And how do we fit all this in, right? Because if we looked at all the DNA that existed in our bodies, it would have enough DNA, if you connected it all, to go to the moon and back 300 times. That's a lot, okay? So how do we fit all this in, right? And so basically we have this DNA double helix that then gets wrapped around these nucleosomes. And a nucleosome is 8 histone proteins in the DNA that's around it. So we could put a circle around here and call this a nucleosome. So that's important to know. And then what we do, or what your body does, I should say, is that we take this little linker DNA, or this piece of tape called an H1 histone. You don't need to know this name right here, H1 histone, but you should know that with the tape and the nucleosome, then we call it a chromatosome, okay? Then we take those chromatosomes and we coil those and we wrap those around into this little fiber. Look at that, it's 30 nanometers across. And then basically we start coiling that on itself, okay? And then that gets coiled on itself, and it continues to get coiled and coiled and coiled until we get our final chromosome here. All right, so things to know: DNA double helix, know your base pairing, know what DNA is, know a nucleosome, know a chromatosome, and know that it takes a lot of winding and wrapping to form a chromosome. Again, this is just a refresher on your base pairing, so make sure you remember sugar phosphate backbone. We have our bases, and that those bases link together very, very specifically, so make sure you know what your base pairing is. Okay, then if we talk about our central dogma, you learned this in 181, right? So first we have DNA replication, so when cells are splitting, we need to replicate all of that DNA. When we are coding for a protein, or when we need a protein, we have to open up that DNA to the specific genes. We have to transcribe a piece of mRNA, that mRNA leaves the nucleus, goes to a ribosome, where it is translated using transfer RNA and amino acids, and we create a protein. That protein then goes to the rough endoplasmic reticulum, to the Golgi apparatus, and then it's distributed around through the cell. So that brings us to the end of this lecture. We've gone over all of the organelles, the cells, and our nucleus and our plasma membrane. So spend some time, make some flashcards today, review this material, and next week we'll start talking about transportation across the plasma membrane and how that's going to actually work. This is really when these concepts become a lot more applicable to later lectures and to sort of the overall functionality of our bodies. So thanks for tuning in. Good luck studying, and I'll see you next time.